Question number 161. If in a plasmid PBr322, the sequence which starts from 5' end as GAATTC to 3' end appears at same sites, how many restriction fragments will result if we use BAM-H1 for restriction digestion? And the options are 2, 3, 4, 1. And we know that the site given in the question, that is 5' to 3' as GAATTC, is the site specific for ECO-R1. So it is this ECO-R1 which will recognize this palindromic sequence GAATTC. Now the question asks that how many restriction fragments will result if we use BAM-H1 for restriction digestion. The unique site for BAM-H1 is present in PBr322, which is here, and therefore circular plasmid gets linearized, giving one fragment. So if we will cut this PBr322 by using restriction enzyme BAM-H1, we'll get one linearized fragment of DNA. So the correct answer to this question is option number 4, 1. 2, 4 and 3 are the incorrect options. So the correct answer to this question is option number 4. Let's discuss our next question. Question number 162. Entry of DNA in a competent animal cell is due to the use of which ingredient or condition? And the options are heat shock at 42 degrees Celsius, cold treatment at 4 degrees Celsius, cold shock with calcium salts, and lysozyme. And the correct answer to this question is option number 1, heat shock at 42 degrees Celsius, because cells can be made competent by treating them with ice cold calcium salts. So this cannot be a correct answer because this step will lead to the formation of competent cells, and these competent cells are allowed to take up foreign DNA by which step? By heat shock at 42 degrees Celsius, which will form transient pores in lipid membrane and allow the entry of foreign DNA into the host cell. The next option is cold treatment at 4 degrees Celsius, and this is an incorrect option because cold treatment with 4 degrees Celsius will lead to the closure of these transient pores which is created at the time of heat shock at 42 degrees Celsius. The last option is lysozyme, and this is an incorrect option. Lysozyme is an enzyme which is used at the time of DNA isolation from bacterial cells. So the correct answer to this question is option number 1. Let's discuss our next question. Question number 163. Downstream processing after a successful run of batch culture does not include which step from options given below? And the options are separation of the protein product, purification of the protein product, expression of the protein in heterologous host, and last option is preservation of desired protein product. And we know that downstream processing involves separation of the desired product, purification of the desired product, as well as packaging and preservation of the desired product. So option number 1, separation of the protein product, option number 2, purification of the protein product, and last option, preservation of desired protein product, are the incorrect options because these steps are involved in downstream processing. Whereas third option, that is expression of protein in heterologous host, is a correct answer to our question because it is a step which is involved in upstream processing. So the correct answer to this question is option number 3. Let's discuss our next question. Question number 164. Example of glyphosate resistant crop available in market is, and the options are Flavor Saver, BT Cotton, Golden Rice, and Roundup Ready. And the correct answer to this question is option number 4, Roundup Ready. Glyphosate is a herbicide whose market name is Roundup, that is aimed at killing weeds. Crops are made herbicide resistant are called Roundup Ready as they can continue biosynthetic process even in presence of glyphosate. So the correct answer is option number 4. Let's discuss rest of the options. The very first option is flavor saver, and this is an incorrect option. Polygalacturonase activity is altered in flavor saver tomatoes. Next option is BT cotton, and this is an incorrect option. BT cotton is an engineered corn plant which are resistant against infection by insects such as Bacillus thuringiensis. And the third option is golden rice, and this is an incorrect option. Golden rice is rich in vitamin A. So the correct answer to this question is option number 4. Let's discuss our next question. Question number 165. Nutritionally more balanced human protein, alpha-lactalbumin, enriched milk was obtained from. And the options are Tracy, the sheep, Dolly, the clone, Rosie, the cow, Polly, the goat. And the correct answer to this question is option number 3, Rosie, the cow. Rosie was successfully developed in 1997. And we can obtain alpha-lactalbumin enriched milk from this cow. Let's discuss rest of the options. The very first option given is Tracy, the sheep, and this is an incorrect option. Tracy was developed to yield alpha-1 antitrypsin. Next option is Dolly, the clone, and this is an incorrect option. Dolly was not a transgenic animal, it's a clone, and it is not used to obtain human protein enriched milk. The last option is Polly, the goat, and this is also an incorrect option. Polly is a transgenic sheep, and we cannot obtain human protein enriched milk from this sheep. So the correct answer to this question is option number 3. Let's discuss our next question.